and I will also post the assignment, the third assignment, which is going to be a programming assignment in a couple of days. And as I said, it will be about analyzing a protein structure doing by reading its PDB file. So you'll learn how to parse, and you'll write a program to parse and get the atom coordinates from the PDB file and do some analysis on that. All right, so last time we were talking about protein structure prediction, and here we have seen three main approaches to protein structure prediction. One good question could be, what is, for example, AlphaFold doing in terms of these three methods? Is AlphaFold a homology modeling program, or is it a threading program? I didn't take a look at that myself, actually. It will be, if you read the AlphaFold paper, you'll see probably it may be a combination of those things. As a deep learning model, it probably uses some homology modeling in it because it's based on extensive training data. It may also have some protein threading parts, because if homology modeling would not work if we have a sequence similarity to a known sequence structure, known sequence with structure. Now we have mentioned ab initio techniques, which basically we told that they can only work for small proteins of small size. They don't make use of previously known structures, and they make use of physiochemical properties and try to optimize certain things like hydrophobic contacts. In homology modeling, let's continue with homology modeling steps quickly. As I said, we are not going to go into details of these methods, all these steps. We're just going to go at a high-level overview, but just know that existing tools model these protein structures with one of those general methods. And in homology modeling, the first step is finding protein by just sequence similarity. You first do a sequence search by using BLAST or FASTA, and if you can find a sequence of at least 30% identity to your target sequence, and if this other sequence has no structure, which means that you have to conduct your BLAST search against the protein data bank as your database. So protein data bank contains proteins with no structure. Therefore, you will have your template model. So this becomes your template protein. And there may be multiple hits. For example, what happens if your protein is similar to one protein structure with 30% identity, another with 35% identity, another with 33% identity? So BLAST is going to get a number of hits. So the second step is actually these are your template proteins that you get a hit for a certain sequence identity to the target sequence. You multiple align them. So this is where multiple alignment gets into picture as well. We multiply align those similar sequences to identify maybe they may contain common parts. For example, if multiple alignment shows that this 30, 33, 35% is all coming from the same region within those protein sequences, it may indicate something conserved in those regions. And if we know the structure of those conserved regions, it will then serve as a good template, as a good starting point to start modeling our target protein. The third step is we first build the model of the protein backbone, which is composed of those alpha carbon, nitrogen, and the carbon. And taking into account the backbone of the template structures. After we have these template structures, after the multiple sequence alignment, as I said, we may identify conserved subregions in those template sequences. We use them as a model to build the backbone. And after the backbone is built, the connecting parts, the backbones may be in chunks. It may not be as a single connected polypeptide sequence. Therefore, there may be some gaps in the chain. Then we model those gaps by using chemical properties, or whether we know the amino acid types in those connecting regions. We see how most efficiently, we may use those amino acids to bridge those gaps. Okay, so there is a loop modeling procedure, which I'm not going to go into detail, but if we have gaps in our templates, there's a loop modeling procedure to substitute those segments with appropriate length by using the amino acids in our target sequence. And after the backbone is modeled, the side chains are added to the model backbone based on the types of the amino acids we have in the sequence. We know that residue R part, that's side chains, are then inserted into the structure to get a more detailed view of the structure. And at the end, there may be small tunings, small modifications we may make to the structure. There are tools like what if or what check. Let's see whether these tools still exist. What check protein. So the what if web interface. So this one, it's a protein verification tools. If you zoom in, as you can see, you submit a protein PDB file to this, and it will then, so should they use the structure an everyday situation in a crystallography lab. So crystallographers are defining a structure. They would like to know what they're doing. Is the overall trace reasonable? Trace means here that backbone, or sometimes called the carbon alpha trace, which is like a three-dimensional curve. It's a curve in three-dimensional space. So is there a shift in this helix? Do all the geometries look expected? So it's basically looking at the distances, bond lengths, whether the bond angles are possible, viable, whether certain side chains can fit into that space. So this is what so it's actually this Ramachandran plots were back in 1963, it's that old. So this is, what check is some kind of a verification tool for your structure. It will tell whether there are certain variations that are not acceptable. So it's like an expert check, a point by point check, whether that protein is viable or not. Okay, so this is what the evaluation of the structure basically means, that whether such a protein structure, after the modeling, whether such a structure is feasible physically or not. If not, there may be certain perturbations you have to do to your protein structure. This is what is done in step 6. So that's a very general overview of the steps of the homology modeling pipeline, which is implemented by Swiss model. Yeah, the Swiss model, EasyPred, there are some existing services in which you don't need to know what they're doing in the background, actually. They're just like black boxes. You just submit your sequence. A couple of days later, you receive, maybe a day later, you receive an email with your models and understanding. There may be different versions of the model that they build. So that's another thing. If I have these different results as predicted models, which one is the correct one? You may never know, but you may find the common parts of those predicted structures as a good starting point. For example, you may use them, whether it's going to, you may, if you are designing a drug, you may check whether the drug is going to interact with these protein structures by using protein docking techniques. Now, the last structure prediction tool that I'm going to mention is protein threading. And I started talking about this on Monday, which I told you that I call this Cinderella method, because we try to have something, two different types, for example, a shoe and a foot. These are two different objects. We try to fit them together and try to then understand whether this shoe is going to fit in that foot. Similar in protein threading, our goal is we have a sequence. We ask whether this sequence is going to have that particular shape. But that sequence could be threaded, could be overlaid onto the structure in very different ways. So it's it's a problem itself. Threading is like a sequence alignment. It's a problem by itself. So we try to align a given amino acid sequence onto some three-dimensional structure. This is the concept of threading. And what we do is, the idea is we have the shapes in the structural universe. We assume that we know quite many of them, and we have a structure library which may be composed of around 1,000 protein structures. So protein structures, general shapes. And a good starting point for that may be the SCOPE database, SCOPE protein database, which we have seen earlier. If we go one level down, and for example, all alpha proteins, if we go one level, you see that there are 455 entries at the next level. So, and this next level actually classifies proteins in terms of their overall shape. And if you look at how many folds are there in all these four classes, 455 here, 255 here, which makes around 700, another 200, make it 900, another 500, make it 1400, so 1400. So if you, these are the major folds, by the way, we can ignore that small protein part. So there are around 1,400 or 1,400 different shapes in nature. If we believe that the protein data bank covers the structural universe nicely, there will be 1,400 different food that back to the analogy of Cinderella, our sequence is our shoe. And we try to fit that to one of those 1,400 candidates. And we are going to try different ways. There may be the difference from that analogy is that there are different ways to fit our sequence onto those structures. First, we are going to, given a specific structure and given our sequence, we are going to find the best way. Let me get my protein sequence here. So given our sequence and given a certain shape, we are first going to find the best way to align our sequence, to thread our sequence onto the structure. It may be this way, or it may be that way, or as I said, it's an alignment problem of sequence structure alignment. After we do that, we do the same thing for all of those 1,400 template structures. Whichever one gets the best score will be used as template for our structural modeling. Some of the steps after we find a template structure, the rest of the steps may be similar to what we do in homology modeling, but that's the basic idea. So it's a certain premises saying assumptions that the number of unique structural folds in nature is fairly small, that 1,400. We assume that 1,400 different shapes are not going to change that much if we solve the structure of all the millions of proteins in CIVISprot or UniProt. We assume that it's going to be one of those 1,400 shapes. And we can actually take a look at how from time to time these folds, new folds are being found, or this database enlarges in terms of fold, but we see that it doesn't grow that large. So it means that new proteins that are experimentally solved each day is going to fit in one of these folds. Okay, so here's there are some description, some textual description of the shape. So how do you describe a protein in terms of its shape? And here's text with just some text. Each of these small sentences below is a textual description. 4 helices in the structure, bundle, they're bundled up and down, anti-clockwise topology, closed or partly opened, left-handed twist. Some of these, they don't mean anything to me. So 3 helices, bundle, up and down, clockwise topology. So it has they have a certain textual terminology to describe that 3-dimensional shape. But when you see it, when you see some of those inside this family, so there are superfamilies also under the fold. All these superfamilies, they have similar shape. Fold is their main shape. Superfamilies are formed with respect to their sequence similarities. And Uh, this is at the lower level, we have domain, families and domains. And if you click on some of those, as you can see, there are links to some representative structures, one SR9. If we click on this, let's take a look at represent the B chain, and the shape looks like this. So this is that 3-helix bundle, domain only, domain in chain, domain in PDB. So yeah, they also have nice three-dimensional visualization of that chain. So this is that shape that they're describing. So scope database actually is a good, the folds in scope database may be a good place to have a threading, build a threading structural library. So I already mentioned the concept of threading. We align or like a place, a sequence, a query, a target protein sequence is placed onto a shape in some optimal way. And then the good alignment gives approximate backbone structure after this point, and we just compute some energy function. When we try to align our query sequence against a template set, for each of these template set, just like that WhatCheck program does, we just ask ourselves whether such a structure is feasible. We compute some energy functions in that case, and again, it involves lots of biochemistry, biophysics, so I'm going to skip those parts. I'm just going to give you the overall. So how we place a sequence to a template. Here is an example. So this is our sequence. Let this be a template structure. We'll just say that let this amino acid be here. Then we know the amino acid sequence. So since the structure also has an order, right? The structure is not just three-dimensional shape. It's a three-dimensional curve like this. So if we put M here, we put that here, we try to just here, the challenge is that we may have some gaps. What if this is not the exact structure, if we have some changes? So we try to say that, okay, maybe this amino acid is fit here, but the next amino acid, let's continue fitting it here. So just like sequence alignment, we may be allowed to put certain gaps and mismatches may be allowed, but what we're basically doing is starting this, or we may start our alignment not from M, but we may assign K to this part of the structure. Okay, so there are lots of degrees of freedom when we're trying to put this sequence onto the structure. But one thing that is not free is that we have to keep this amino acid order when we are matching amino acids to certain positions on the structure. So this is the alignment is done by assigning these amino acids to the peptide. So as I said, it also has a certain order from N terminus to C terminus. So they are placed. Each of this placement gives a 3D position to these amino acids. So this template structure allows us to give positions to these amino acids. But since there are multiple different ways, we try to look for the most optimal way to do the assignment. When I say optimal, how do we say, for example, what is our mismatch or what is our penalty function for placing an amino acid here? There may be some global functions. For example, if your amino acid is hydrophobic, which means that it doesn't want to be at the surface, placing an amino acid at the surface of this template protein structure should be penalized, right? You don't want a hydrophobic protein at the amino acid at the surface of this protein structure. So maybe you may give better scores for hydrophobic amino acids that are matched to positions on the structure that are at the core, that are not at the surface. So there are some global, so this is basically an energy function that you're computing, whether it wants to be at the surface or not. And there's another thing is, for example, two amino acids that are far away, like this K and V, K and A, that are far away in the sequence of, if you make this assignment on the structure, they become neighbors in the structure. We may compute a score or a penalty function saying that how preferable to put these two amino acids together to make this assignment. So by considering all possible combinatoric ways of assigning these things, we find ourselves an alignment, sequence versus structure alignment, which minimizes this total energy function. So the smaller the energy, the better, the more stable that assignment is. As I said, there are different measures, whether two amino acids want to be close to each other, whether this is a global feature, whether an residue fits the structural environment, like whether it's the surface, whether it's the core and this type of this amino acid. Valine, does it want to be in the surface? If not, we give a penalty there. So we may, at the next step, we may try to make the assignment to another one here. So as I said, this is the part that I'm not going to go into detail. There's a very complicated alignment algorithm that considers all these different matches. And this, by the way, protein threading itself is an NP-complete problem. It's difficult. It's not like as simple as pairwise sequence alignment. Just think about combinatorically assigning each of these amino acids to all possible positions here. Although you need to maintain the sequence order, there are still infinite millions of different ways you can make these amino acid position matching. And this is what we try to establish here. We match each of these amino acids to a position in this template structure. If this template structure is different, if you try to do the same thing for a different structure, like this, for example, if you try to align your sequence against this structure, you would get a different score. If you align it against this one, you get a different score. So we do this for all of the proteins, template proteins in our template database, which we have seen that in scope database. If we use the fold level structural classification, we will have about 1,400 such different templates that we have to compare our sequence against. So these methods, therefore, protein threading, you cannot do it on real time. When you submit your sequence to a protein threading server, getting a result from their server may take 2 days, 3 days. Most of the time, these are like jobs that you submit, which you wait a couple of days until you get some modeling result. So it's really time consuming. And in those servers, they don't, there's also a limit that those, the computational resources are limited. If there are many users submitting jobs to these servers, you may even wait longer. But that's basically the idea. We compute a score function based on a particular assignment of these amino acids to these structural positions. So I hope this slide clarifies how a sequence can be put onto a structure. Okay, so and when we have gaps in our sequence, we also penalize that. So gaps are penalized. These amino acids being here are you're welcome, by the way. So this is a good question that you asked. So this, what you're asking is basically the algorithm of threading. And I can, there are some nice seminal protein threading papers. Protein threading. One of them is like, that shows it's NP complete. I think this one, yeah, by Lathrop. It's a 1994 paper. And you can also take a look at the paper, you can look at the papers that cite this seminal paper. The protein threading problem with sequence amino acid interaction preferences, which is, so this one, sequence amino acid interaction preferences. What does this tell you? This keyword should ring a bell. It's this one. Look at this example, K and A. What is this? K is interacting with A in 3 dimensions. So if you take this interaction preference into account, which means these are non-local interactions in sequence, then the combinatorial complexity really explodes and it becomes an NP-complete problem. An NP-complete problem is it's a set of problems which are very difficult to solve. Like one of them is like prime factorization, which is, so it's difficult. Therefore, that 256 bits hashing. Therefore, that's why mining for bitcoins is difficult, because it takes a lot of computational power to do that. So that prime factorization problem is a difficult problem. And this one is saying that this protein threading problem is as difficult as that one. So it's in the same complexity class. So I think this, do I have the, can I read this paper? Is it the only abstract? Where is the full text? Share favorite sites, display options. I think I need to click on this one. This is just PubMed link. If we have, as Middle East Technical University, subscribe to this journal Proteins, I think we'll be able to get the full text as well. Permissions, I think we don't have that as a digital version, the PDF of this article. Anyway, I think I have that in print. So this was one of the papers I read during my PhD. So I have it as in print. So if you're interested in this, I can share this paper with you. If I dig in my list of papers that I had read earlier, I will be able to find this paper and share this with you. Anyway, so this is a difficult problem by itself. This is basically what it's saying. So we first build a template database of all known shapes in nature. Yeah, I will share this paper on O2 with the whole class. I'll do that and scan it and share with you. Or maybe I can find a PDF directly on the internet. But yeah, I have that paper and I will share it. Oh, so you already shared the link. Thank you, Ahmed. Let's see whether it's that paper. Yes, that paper. Yeah, it's this paper. So you already found it. So it's good that this video is also being recorded, which means that it's nice that everybody's going to, who's watched this lecture video, is going to have access to this paper. Thanks for sharing it. So yeah, Google most of the time works on these issues, so it's good. So this is the paper, original paper. So let me download this, and also I will share this with you also on O2. All right, so this PDF reader, what is this? This is not Chrome or anything. Is this some Microsoft? Oh, this is Firefox, I think. Yeah. Let me put it here, which is a better built-in PDF viewer. And yeah, I know where its download button is located. So I got the paper, so I will also share this with you on O2. Yeah, it's an MP hard means as difficult as this. MP complete does not mean that it's harder, or MP hard doesn't mean that it's harder. MP complete means that if somebody gives you this solution, it's easy to verify if the solution is correct or not. So this is now outside the scope of this class, so I'm not going to delve into this issue, but yeah, it's the threading problem itself is really difficult. But what we do is we first build this template library, then our scoring function gives a score to that alignment, and then we assign our confidence whether that alignment is good or not, whether that sequence prefers that structure or not. So basically we find the foot that best fits the shoe. This is what is done for this confidence assessment level. And after that template doesn't, after we find the foot, it doesn't mean that it's the exact structure. Just like again in homology modeling, we may do small improvements on that template. We may use it as a starting template in homology modeling and continue the steps that we do, like loop modeling. Since we may have gaps in our threading, we may try to work on those gaps. We may work on the side chains, do some tuning, improvement on the final structure, and that becomes the predicted structure. So that's what threading is. And here are some results that prediction of some protein structures by threading. So if this is the actual, this is the predicted structure by threading. They are really similar, right? This is the actual structure. This is the predicted structure by threading. So these are really good examples. Just like looks like the results by AlphaFold. Now that's actually this is going to bring us to our next subject. If I give you these two structures, for example, this is actual, this is predicted. This is the actual, this is predicted. For example, is this result better than this result? So how can you evaluate whether actual and predicted, these two structures, are really similar to each other in shape? Right now, we're just looking at them and see, okay, they seem similar, their shapes are similar, but how good is that fit? That will lead us to our next subject, which is structural alignment. Structural alignment problem is the problem of trying to again match two structural parts with respect to each other and superimpose those match parts so that they're as close as possible, and measure the closeness of those superimposed parts. And one thing that we are going to see is that the RMSD, root mean square deviation measure, which tells us how really similar those structures are in terms of their shape. So structure, in other words, in order to evaluate whether the predicted structure is close to the actual one, we will now do structural alignment of these two things. And the structural alignment result, if it aligns well, and if after the alignment, if the superimposed means that we can, in 3D space, we are free to move and rotate the predicted structure, right? I mean, they're just shown side by side, but we can just show them on top of each other. If you remember the AlphaFold blog, the predicted and actual structure were not shown side by side. They were shown on top of each other, superimposed. And this means that they were aligned and superimposed on top of each other to show how similar they are. After superimposition, after the best way to superimpose them, they may still not be exactly on top of each other. The deviation between those match parts, the average distance of those average deviation is called the RMSD measure. Let me write in the chat box, which we are going to see, root mean square distance, or root mean square deviation. So we get the distances, we square them, we take the average of all the deviations from different parts of the structure, and we take its root, and its unit is angstroms. So since our protein structures, the measurement unit is not meters, centimeters, millimeters, or microns. We are at the angstrom domain, at their atomic level, so it will be angstroms. And two structures with an RMSD of 2-3 angstroms, it means there's a really good match. Okay, 2-3 angstrom is good, but if you have 9 angstrom difference on the average, 10, it means that the structure are really different. And this is a bad example of threading. So due to these 3 helices, it made this match. This is the actual structure. This is the predicted structure, which has some obvious differences. This has, this is like a longer structure that is predicted. There are some existed prediction programs, Threader, Fugu, Prospect. Actually, I recently did some threading, or was it homology modeling? I think it was homology modeling for a friend of mine using a server. If I find those, I will take a look at my emails, and I will also share that program with you. Okay, it's linked. So if you're going to do this such kind of a structured prediction later, you may refer to those programs. Okay, one of the reasons sometimes we do structural prediction is that, for example, when we have a protein structure, for example, which is truncated at the end, if we have a protein structure which is missing some 20-30 amino acids at its end, how is going to how this truncation is going to change its structure. You may do that, for example, first trying to model that truncated protein sequence, and then afterwards you can compare the non-truncated version and this one, whether certain active sites are lost or whether the surface of the protein changes and things like that. So this is usually what is done. We usually in bioinformatics or in biology, if you're comparing things, a normal case versus an abnormal case, wild type versus disease type versus treated, or whether there's truncation or not. So, and how that changes the biology, the biological function of the things that we analyze is basically a general way of analyzing things. So yeah, I mentioned CASP earlier. So AlphaFold was the winner in the last CASP by a big margin. It was such a big margin that the organizers of CASP, I mean the initiators of CASP in the 1990s, they now consider the protein structure prediction problem solved. So the CASP competition, which was held every 2 years, it first starts by collecting some template benchmark structures from structural biologists. So they determine the structure experimentally. So they probably are in close contact with X-ray crystallographers or people who have cryo-EM in their lab. So, and they determine the structure of a novel protein, which the structure of that protein is not deposited in the protein data bank yet. So this is the very difficult part. It takes a lot of months to get structures, secret structures, whose sequences are given to the organizer or organizers, and then they give the structures to organizers too. So these are structural biologists who give the challenges, the tasks to whoever is going to compete in this competition. And there are predictors, which come with prediction programs like threaders, homology modelers, and their goal is to compete in this competition to predict the structure from sequence. And afterwards, the predicted structures are compared to these actual structures by using the structural alignment algorithms that we are going to see next. And then the results are analyzed by the referees. There's a committee, a jury, and they have a conference at the end of after the results are announced. They have a conference to discuss the results, which algorithm did best. And there are also subtasks. For example, prediction of just secondary structures, or prediction of just the contact points between two chains of proteins. So there are different subtasks in CASP as well, but the main task, which is predicting structure from sequence, is what everybody most of the time focuses on. All right. So there is also the CAFA, completely automated prediction of structure prediction. And in CASP, they were actually, some of these results may be manually. The CASP predictor may make some of these models, may correct some models manually before submitting their results. But there are also parts of CASP in which fully automated programs compete with each other. So these are some statistics from the 6th CASP competition. All right. Now we mentioned SCOPE, which provides a classification of structures. Now I'm going to talk about, so I already discussed the SCOPE database a little bit. There are also different protein structural databases too. Now we're going to talk about structural alignment algorithms. And I will mention DALI in detail. And during my PhD studies, I even developed a tool for structural alignment, which was called CTSS, which is an acronym for curvature, torsion, and secondary structure. So it was a structural alignment method which uses these three information, treats the protein structures as a three-dimensional curve in space, and by computing local curvature and torsion values at these amino acids, using those three values and secondary structure information, using these three values to build, and just using pairwise dynamic sequence alignment to try to align the structures, because curve and torsion, these are like shape features which are local but invariant to translation and rotation. For example, DALI uses something else. There is this CE algorithm that uses something else, but the problem that we're given is this. Let me try to draw it here. We have some protein shape, and we have another protein. Okay, maybe something like this. Our goal is, as you can see, their lengths are different. This may contain M amino acids. This may contain N amino acids. Our goal is try to find maybe some local subshape. For example, this part. Let me draw it in red. This part may be similar to this part. Okay, so find a substructure or as large as possible, which is similar to in shape to this one. And after you find these shapes, for example, if you were to move this black region, so let me actually try to do that like this. Let me select this part and try to move it here. It's not transparent. And also, if you rotate it, for example, transform tools, let's try to rotate it. Maybe if you rotate it like this and then move, you'll see that it will maybe fit better. So if I do it like this, are they going to be exactly the same? No, there are some differences. So after you find actually finding the match part first is a difficult problem. Are you going to match this part to this or that part to that? So finding the matches is the difficult part in the structural alignment problem. And after you find, you decide that this part is matching to this one, the thing that I just did, moving this over here and rotating it such that they are on top of each other as much as possible, is called a superimposition. And superimposition is a solved problem. If somebody gives you the matches of points or matches of certain parts, translating and rotating it in the most optimum way, so that the distances between those match parts are minimized, is a solved problem, which can, there's a very efficient algorithm which works in linear time. When I say linear, this is like a point cloud superimposition problem. Imagine we have a set of points. Let me try to draw them like this. Okay. I name them like this: 1, 2, 3, 4, 5, 6, 7, 8. Okay, now I will give you another 8 points: 1, 2, 3, 4, 5, 6, 7, 8. Okay, 1, 2, 3, 4, 5, 6, 7, 8. Now, here's the problem. Considering these points, just actually you don't need to, and their orders. If I tell you that each corresponding number is matched against the other corresponding number, so I here is matched against the other I here. Okay, 1 matches 1, 2 matches 2. Now, what is the best way to move these 8 points on the right hand side? What is the best way to translate them? Translation means just this. So after you select, for example, here, oops, I'm currently drawing. So translation is just panning. Okay, so this is, oops, I didn't, after I select, I didn't say Control X, Control X. So after I select it, oh, is it a different level? moving it means translating, and rotation is also obvious what it means. So if you're allowed to only translate and rotate these sets of points on the right hand side, what is the best translation and rotation? That is going to minimize this, the distance between the ith point in the first set. Let's call this set A, let's call this set B. I find the distance between AI and BI. Okay, I take the square of these distances. Okay, root mean squared distance. So these are the squared distances. I sum them up, and so I take the mean. There are 8 of them. So this is the average square distance, and I take the square root of this thing. Okay, the very important thing here is that the distance is computed only between matched points. Okay, so the problem that I ask you is minimize this. Find the translation and rotation such that you're going to move these points over to here, you're going to rotate them, and at the end you're going to minimize this, which this measure is called RMSD. And the minimum value is going to be something like, for example, 1.2 will be the minimum distance between them in some units. Or if this is a space in angstroms, there will be the distance between these 2 points, between 8 and 8 will be some distance. And if you minimize it, and if you find it to be a value of 1.2, that shows you something about how close these 2 point sets are in terms of their shape. For example, if you consider them as a curve like this in 3D space, it will tell you how these curves are similar overall. All right. So, and by the way, this finding the minimum RMSD, finding the minimum translation, best translation rotation matrix, given the corresponding point correspondences, as I said, is a solved problem. The optimum one, RMSD, can be found in linear time, where n is the number of corresponding points. Okay, so this is done by using a singular value decomposition of a 3x3 matrix in 3-dimensional space. You have the XYZ coordinates, so they first look at the covariance of these coordinates of these points. That covariance matrix is the 3x3 matrix, and the singular value decomposition of that gives you the translation and rotation. So that's really like a magical algorithm that works. But just know that I'm not going to go into details of this algorithm. That's enough detail at this point. Just know that it's a solved problem. But this is not the structural alignment problem. Structural alignment problem is dealing with the case that which part I'm going to match with where, which amino acid matches with who. Okay, alignment, actually, if you remember sequence alignment, what it was doing is that there's an A here, there's an A here, there's an A here. If there's a single A here, for example, which one of these A's are going to match this? Maybe none of them. Maybe it's just going to be a mismatch. But alignment problem really is the problem of finding those correspondences. And when you have given 2 structures like this, finding the local alignment, which substructure is similar to which other substructure, is a difficult problem. Therefore, all we have in structural alignment are these heuristics like DALI, VAST, or CE algorithms. And I will stop here. I will talk about the DALI algorithm in detail. So I have only 1 hour to go until the end of the slide. So we will finish this discussion on protein structures on Monday. But I will post the assignment anyway, because we know enough about protein structures. So you should be able to understand the assignment and start working on it right away. It's not the assignment is not going to be about protein structural alignment. It will be analyzing a single protein structure. Okay, so that's it from me for today. If you have any questions, I will be happy to answer them. Let me stop recording, and I will share with you